Hi guys, welcome to part two of Anatomy Physiology lesson 7.07 on the sliding filament theory. So what we're going to do before we start talking about the specific steps after calcium leaves the sarcoplasmic reticulum, I want to make sure you know what parts of the diagram we're going to be using are, and it's going to require a little bit of review. And I'm going to show a 2-minute video on the whole mechanism. We're going to note out the details, and it'll go into the third part to finish noting those details, and then I'll show the video again. So this is the diagram we're using. You'll notice over here to the sides I have redrawn my pieces. If this looks blurry, it's because I took a little tiny picture and I blew it up. So I tried to outline and recolor to make it more visible. If you want to redraw these, go ahead and just pause the video. But what are we looking at here? You should know all of these parts by now. And if you don't, as we're going through this, you need to stop. You need to go back. You need to make sure you know what all of this is. And then you'll have a better understanding of what's going to come. So we left off, this is the T tubule. And we had said that when a nerve impulse gets to the axon, it releases neurotransmitter. It binds to receptors across on the other side of the synaptic cleft. Those receptors cause sodium to flow, and the sodium flows down into the membrane. And when it gets to the T tubules, it causes, this is the terminal cistern. Terminal cisternae. And the terminal cisternae releases calcium. Now this shows the calcium leaving from the terminal cisternae, but that actually floods all the way down the sarcoplasmic reticulum, and we get calcium that leaves, and that's what this little orange dot is. This is calcium. Whoops. Calcium, or Ca+2. So what we're seeing here is step number 6, where calcium leaves sarcoplasmic reticulum. And then what's happening here is step number 7. So we're going to pick up with step number 7. Looks like a golf club, right? That's the myosin fiber, and this is a cross bridge head. Cross bridge head of myosin fiber. You'll notice that it has 2 small circles here, 1, 2. When we have 2 small circles like that, that represents ADP and a phosphate group. When you see a single circle, that represents ATP. And if you remember from biology, and we've talked about other times this year, ATP is the a molecule of energy for our cells, and that's the energy that's going to power this whole cycle. The blue dot is the binding site. And currently that binding site is blocked by this filament. This filament down here is the actin, so the gray beads is actin. And the binding site, and we should say binding site on the actin, is covered by the thin filament here, tropomyosin. Tropomyosin. This complex is called the troponin complex. So what we see, step number 7, is going to be the calcium ion binding to the troponin. That's what's going to happen next. It's going to be, when we get to our main sheet, that's what we're going to see. All right, so before we go into details, let's watch this video, and I'm going to need to share my screen again. There's been lag. See how I do today. Share computer sounds.
Contraction of skeletal muscle tissue occurs when actin and myosin myofilaments slide past one another, causing the sarcomere to shorten. It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules attached at the individual myosin heads, actin, tropomyosin, and troponin. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with the troponin molecules. This causes the tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross bridge. The remaining adenine diphosphate molecule is then expended in what is called a power stroke, when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, adenine triphosphate molecules attach themselves to the myosin heads. These molecules trigger the release of the myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate as the heads move back into their resting position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again, and the sarcomere will continue to shorten until the calcium ions are transported back into the sarcoplasmic reticulum and the muscle relaxes.
All right. Oops. Shoot. Pause the video. Okay, so here we go. This has all of the steps, steps 6 or 7 through 13 in the muscle contraction. So we said we're going to start with step 6, where calcium leaves the sarcoplasmic reticulum. Sarcoplasmic reticulum, which is what's happening here. So here we go, step number 7. The calcium is going to bind to the troponin complex. The calcium is going to bind to the troponin. What that does, and you'll notice our current state here is I have an ADP. ATP has been split, but is still attached to the myosin head. So I have an ADP and a phosphate attached. The binding site is covered, so we'll kind of talk about the mechanics here. So when that calcium is released, ADP and phosphate are bound to the myosin head. The binding site's covered by the tropomyosin. So in step 7, that calcium ion is going to come down and bind to the troponin complex. And what that's going to do in step 8 is, now that this has a calcium bound to it, notice that the tropomyosin moves off of this binding site and exposes it. So this causes the tropomyosin to move, which exposes the binding site. All right, we are going to pause, or we're going to break part 2 here, and I'm going to do part 3 in another video, just to keep the length of time manageable. So I'll see you in part 3.